Beyond nutrients and carbon quality, there are environmental impacts on decomposition. Here, we'll cover the impacts of factors such as moisture and temperature on rates of decomposition. Moisture and temperature are two of the more important environmental factors that control rates of decomposition. Decomposition increases with increasing moisture until soils become so waterlogged that anaerobic conditions inhibit decomposition. So when we think about the first factor, moisture, decomposition rates increase as moisture increases. But then, at some point, you have so much water in the system that the rate of oxygen movement to microbes declines. And as a consequence, decomposition rates can even fall as moisture increases. So moisture has an important role in determining how fast decomposition proceeds. In general, as soils dry, microbes have less access to organic matter and nutrients that are being made available to the microbes. In addition, there's greater osmotic potential of drier soils. So solutes that are present in water become more concentrated, and this causes greater stress on the microbes to maintain a certain water level within them. There are also drying and wetting cycles that happen in soils as rain falls and wets soils up, and then they slowly dry due to evaporation and transpiration. As soils dry out, bacteria often die, and this drying cycle kills off bacteria. When you have a wetting cycle that follows it, it often flushes the contents of those bacteria into soil solution. High soil moisture is also associated with less oxygen. So just as we talked about, if soil moisture is on the x-axis, decomposition is going to increase to a certain point, and then decomposition becomes limited because there's not enough oxygen. Oxygen moves through water somewhere on the order of 10,000 times slower than it does through air. So having a lot of water in there without a lot of airspace really can deplete oxygen levels to bacteria and reduce decomposition rates. As far as temperature is concerned, although microbial respiration and decomposition increase with temperature in the short term, indirect effects constrain their temperature sensitivity over annual to decadal timescales. So there's two factors here that as temperature increases, decomposition tends to go up. But this is more of a short-term effect, and a longer-term effect for the same amount of temperature increase tends to be more muted and sometimes even flat. So let's get through some of the factors that are associated with temperature and why decomposition tends to increase on the short term. Here we have two graphs. The graph on the left shows changes in respiration rates of tundra soils that were incubated in the lab. And on the right is going to be soil respiration rates from across the world. In both cases, and I'll start here with the panel on the left, we have temperature on the x-axis. And the temperatures here start at negative 10 degrees and go all the way up to 35 degrees Celsius. Now on the y-axis we have respiration. And respiration here is measured in terms of oxygen consumption per gram of soil per hour. So we usually think of respiration as carbon respiration, but you can measure oxygen consumption just the same. So respiration here on the y-axis, and this is an index of how much decomposition there is. So this is a pretty typical curve where you have an exponential increase in respiration rate up to a certain point, and then you actually will have a decline. So once you get up to a certain temperature in a given soil, which here appears to be somewhere on the order of 15 to 20 degrees Celsius, you'll actually get inhibition of processes. And you can see that your peak decomposition here is at 25 degrees Celsius, and after that you actually have declines in the rate of respiration. Now this initial part of the curve is an exponential curve. And we can fit a function to that, for example, but for here, let's just compare what happens when we go from 0 to 10, for example, to 10 to 20. Now this appears to be somewhere on the order of 40 right here, and this appears to be somewhere on the order of 120 as far as units are concerned. To go from 40 to 20, This is a tripling of respiration rate. At 10 degrees, if we're at 120, and here we're at 300, this again is almost a tripling. I'll just say it's a tripling. So it's 3 times. So you can see that it's not a true exponential because it starts to slow down right around 15. But if this were to be true exponential, we probably would have been up here. Still, somewhere on the order of a tripling of respiration over the initial part of this curve. And often we'll talk about a Q10, which is the relative rate of increase in a given process with a 10 degrees Celsius increase in temperature. And here, empirically, it's about 3. So when we do a lab incubation and we increase the temperature of a soil and take a look at oxygen consumption or its respiration rate, we can see that it triples with every 10 degrees C increase up to a certain point, and then it starts to level off and decline. That's a short-term increase in temperature and its consequence for respiration. Here we have soil respiration rates. So you go to the field and you take a look at the rate at which carbon dioxide is being released from the soil per unit area per unit time. And again, on the x-axis we have temperature. Here you can see is an exponential increase in soil respiration with temperature. And this might be hard to do, but we can compare what kind of increase we get going from 10 to 20 and 20 to 30. So if we say this is roughly 1 and this is roughly 2 as far as units in soil respiration are concerned, to go from 20 to 30 we go from roughly 2 to roughly 6. So we're talking somewhere on the order of 2 to 3 times the increase in respiration with a 10 degrees C increase in temperature. Now it could be that both of these are 3 and I'm just estimating here. So for all intents and purposes our Q10 might be something like 2.5. So pretty similar to the Q10 of 3 that we got over here. Now the units here just are in micromoles of CO2 per meter squared per second, which is about the same of grams of carbon per meter squared per second. So we're talking in a lot of these soils somewhere on the order of 2 to 10 grams of carbon being released per meter squared per second, and doubling to tripling of respiration rate with every 10 degrees C increase in temperature. But also notice that this is an exponential increase, and we haven't necessarily seen any kind of inhibition of respiration with these changes. So these systems that do get really hot, for example, they tend to have microbes that are adapted to the higher temperature, whereas this tundra soil just don't have microbes that do well at, say, 30 or 35 degrees C, because those temperatures, for all intents and purposes, are never reached. Now, understanding why processes such as decomposition or respiration increase as temperature increases has to do with the activation energy of enzymatic reactions. When enzymes and substrates collide, they'll have a certain amount of energy. And a certain amount of energy is required in order for that reaction to occur. And that's the activation energy of that reaction. Now, as temperature increases, it actually doesn't have a big impact on the number of collisions. So we think of particles moving a lot faster as temperature increases, but really to go from 10 degrees Celsius to 20 degrees Celsius might impact the number of collisions by maybe somewhere on the order of 3%. It's not a big factor. So the reason why as temperature increases, the amount of decomposition or respiration increases isn't because enzymes and substrates are colliding a lot more frequently. Instead, there's a large increase in the energy of the average collision. So those particles have a lot more energy when they're coming together. And the enzyme and the substrate tend to collide at higher energy. And this means there's a higher probability of them colliding at an energy that's sufficient for that reaction to occur. What we've also learned relatively recently is that the more biochemically recalcitrant organic matter is impacted more by temperature. So as temperature increases, all rates of reactions increase. But it's the biochemically recalcitrant organic matter that's impacted more. And the reason is that the biochemically recalcitrant organic matter have a lot more high energy bonds. And that decomposition is going to be dependent on breaking these bonds. And the way that the chemistry works is that you're a lot more likely to have an increase in the energy that allows you to break the double bonds, for example, than the single bonds. So as temperature increases, all rates of reactions increase, but it's really the more recalcitrant organic matter that's impacted more. Now I should also state that there are negative feedbacks associated with increases in temperature. So not the negative effects of increases in temperature as you get beyond optimal levels of temperature for a given organism, but there are feedbacks. There are processes that occur that tend to reduce the increase in response with increases in temperature. So for example, we have something called acclimation. And what that is that if you were to take a look at short-term increases in temperature, you might have a reaction, an exponential increase in a reaction that looks like this. So temperature goes on the x here, and the rate of reaction, we'll just say d for decomposition, increases like this. But with long-term increases in temperature, so if you go from, let's say, 10 degrees to 20 degrees short-term, you get this amount of increase. But as that system stays at 20 degrees, actually the rate of increase declines over time. And this is an acclimation process. And there's a couple factors that are associated with acclimation, but the microbes themselves actually respire at a lower rate than you would have expected from the short-term increases. And this can be due to other limitations, for example, and some of the same things that we talked about with plant production. But you can have acclimation responses or downregulation. So what we often see in short-term responses don't happen in long-term responses, in part because of acclimation responses.